Okay, so we've looked at the simple saccharides. We've looked at the monosaccharides, but we don't really come across monosaccharides in nature very much. Usually, with the exception of honey, of course, which is filled with fructose. But mostly we come across it in disaccharides and polysaccharides. Di meaning two, poly meaning many. And we'll see that there's a wide variety of disaccharides, two. So basically, you take any two monosaccharide units, generally the six carbon monosaccharides, and you link them together with a carbon-oxygen-carbon linkage known as a glycosidic linkage. And this is the glycosidic linkage here. And we also refer to the glycosidic linkage as an alpha or beta. In this instance, it is trans to carbon 6 here. So we would call that alpha. In fact, we would refer to this as an alpha 1,4 linkage because it is linking carbon 1, or the anomeric carbon, of one disaccharide and carbon 4 of the other. And it is a carbon-oxygen carbon bond. So that is an example. Let's look at some more. So there's three common ones, the most common ones: maltose, lactose, and sucrose. I'm sure everyone's familiar with sucrose. You may not be as familiar with maltose. Now the hydrolysis of maltose releases two molecules of D-glucose. So you have the most common monosaccharide. They come together and form maltose. Let's see what link that is. So we have carbon 1, or the anomeric carbon of 1-glucose here, linked in an alpha fashion because it is trans to carbon 6. So when it's in trans, we call that an alpha. And so we would call that alpha maltose. And the linkage would be referred to as an alpha 1,4 glycosidic linkage. So an enzyme were to break that down into its glucose monomers, it would have to be an enzyme that broke down alpha 1,4 glycosidic bonds. And as we know, or we've been trying to be taught this term, enzymes and proteins in general have very, very specific three-dimensional structures. And so if an enzyme can break down an alpha 1,4 glycosidic linkage, it can't break down a beta 1,4 glycosidic linkage, and so forth. And that'll come important in just a second. So maltose. Lactose, no doubt you've heard of lactose. Lactose is also known as the sugar in milk, or milk sugar. So it has one molecule of D-galactose and one molecule of D-glucose. And this time, however, it's a beta 1,4 glycosidic bond. What does that mean? Well, beta, let's look at the linkage. So you can see that the linkage is now cis to carbon 6. It's on the same side of the ring. And so we call that a beta 1,4 glycosidic bond. And so, as I mentioned with maltose, with lactose, there's a very specific enzyme that breaks down that beta 1,4 glycosidic bond. And the problem is, a lot of people don't have that enzyme, and they become lactose intolerant because they don't have the enzyme that breaks that bond. And so what you do is you have a buildup of lactose in the blood. And it's not a good idea to have a high concentration of sugar in your blood, as you're probably aware. It leads to a lot of problems. And so a lot of people don't have lactase, which is the enzyme. We'll find out that enzymes that break certain bonds generally have that ASE ending, which means that they break whatever comes before the A's. So lactase breaks down lactose. And certain adults had that enzyme when they were children, and then it goes away with time. They stop making lactase. And then others were just born without the enzyme at all. And so that is a problem for them. And so either they just don't drink milk, or they buy commercially available lactase and then put it in their milk, or they can buy milk without lactose in it. Another problem with lactose that people have is that we know that glucose is the preferred monosaccharide for your body to break down and get its energy from. So galactose generally gets converted to glucose for the metabolism to take place. Galactose doesn't actually get broken down. It gets converted from galactose to glucose. And so you have an enzyme that does that recognizes galactose and then changes the structure around to glucose. Now, people who don't have that then get a buildup of galactose in their blood. And any buildup of any sugar in blood is a bad thing. And so that particular disease is called galactosemia. And it's much, much rarer than the lactose intolerant, but it does exist. And it's a real problem because you come into contact with galactose from a wide variety of sources. So then we come to sucrose. Sucrose is the most common that we certainly come across. And it has a really unique linkage because it doesn't go 1,4 like most other disaccharides do. It actually has an alpha-1 beta-2 glycosidic linkage. So what does that mean? So here we have glucose, and it is connected to fructose. So it's one glucose, one fructose linked together to make sucrose. So the anomeric carbon on sucrose, as it is on all sugars, is on all sugars that have a I need a drink there. On all sugars that have an aldehyde or aldo sugars, the anomeric carbon is going to be number 1. So it links to fructose, but fructose is linked to carbon 2. It's fructose's carbon 2 that it's linked to. And we call it an alpha beta because on the glucose here, you can see that this bond is trans to carbon 6. And so this is alpha linkage to the glucose. However, on the fructose part, on the bottom part, this linkage is now cis to carbon 6. So it is beta. So we call this alpha 1 because it's linked between alpha 1. It's linked between carbon 1 of the glucose and carbon 2 of the fructose. And it's alpha on C1 because it's trans to its C6, but it's beta on the fructose because it's now cis. And this is a unique way of putting together a disaccharide. And so the enzyme that breaks this particular bond is different from the other two because now we have a unique bonding that wasn't present for either maltose or milk sugar. And so we have, or lactose. And so we have an enzyme called sucrase, also known as invertase. It was called invertase because this linkage is inverted from most disaccharides. So it was called invertase for the longest time. But now to sort of try and get it together with the common names for everything else, we call it sucrase because it breaks down sucrose. So let's look at polysaccharides. These are really large polymers. composed of like the tens for like oligosaccharides to many, many thousands of monosaccharides, all joined together by these same glycosidic linkages. So they do not taste sweet. They are non-reducing. They don't have an aldehyde or keto group to reduce. So the three most abundant polysaccharides are starch, glycogen, and cellulose. You're probably familiar with starch and cellulose, maybe not so familiar with glycogen, unless you're like a long distance runner or something. So these are referred to as homopolymers because they're all made of glucose. So it's the same thing. They're just glucose linked together in different ways. So when enzymes break down any of these polysaccharides, the product you're going to get is glucose. You're all going to get the same. Heteropolymers may contain all sorts of other things. They could have sugar acids, amino sugars, non-carbohydrate substances. They could be phosphorylated with, they could be have sulfur groups, sulfate groups, all sorts of different things. But these particular polysaccharides are homopolymers that are just made from glucose, but put together in different ways. So starch is the most important source of carbohydrates, like potatoes, pasta. They have a lot of starch in them. And they really account for about half of all the carbohydrates you ingest. It comes from starch. And it's a mixture of two different polymers. One is called amylose, and that is just a linear structure of sucrose joined together by alpha 1,4 linkages. So here's carbon 6. There's our anomeric carbon. There is the linkage, and it is in trans. That is an alpha 1,4 linkage, and it just goes on and on and on and on and on. So it's just one long stretch of glucose units. Amylopectin, on the other hand, is a branched polymer. It comes off these structures with a different linkage. So amylose gives you this sort of spiral structure because of internal hydrogen bonding, sort of keeps it together. So you get this long, long spiral of D-glucose chains. Now, where the bonding is different for amylopectin, is instead of alpha 1,4 linkages, you get these branching off from 1,6 linkages. So here is our carbon 1, and there's our carbon 4. So that is the alpha 1,4 linkage that keeps the long chains together. And then you get a branch where carbon 1 from one glucose is then linked to carbon 6 in the long chain. And so we call that alpha 1,6 because that is also still trans to the carbon 6 on this particular glucose that's coming in. So then that branch will go off with alpha 1,4 linkages as far as it can. And so the long chains are held together with alpha 1,4 linkages, and the branches are put together with alpha 1,6 linkages. So glycogen is the sort of energy reserve that we have for sugar, like short-term sugar storage. It's quite similar to amylopectin, although it's much more highly branched. There's only 8 to 12 glucose units between branches, and the branches are shorter. So it basically is like a really fast, short-term storage for sugar. And like distance runners will like carbo load before a race. What they're doing is they're building up there's storage of glycogen because they're going to use a ton of energy running 26 miles. And fat, which is also energy storage, is just going to take too long to break down. That takes hours to break down. You need something in minutes. You need something in seconds. And so you need, like, I need my sucrose, I need my glucose, and I need it right now. So for that, you would use glycogen. And it can be broken down into its D-glucose subunits by the same enzymes that break down starch. So starch can break down alpha-1,4 and alpha-1,6. Those are the same things that are present in glycogen, so you can just break those down pretty quickly. Now, cellulose is also just glucose because all these three are just glucose. It's fibrous. Why is that? Well, because it's a linear polymer of glucose, and the linear polymer groups sort of stack on top of one another with hydrogen bonds. And they're held together with beta-1,4 linkages. And so we don't have a lot of enzymes to break down beta-1,4 linkages in these long chains. We can break them down when they're in short disaccharides, but polysaccharides, we don't really have an enzyme that can do that. And so we can't digest cellulose. Termites can. Termites have enzymes that can break down long chain beta-1,4 linkages. And so to a termite, a piece of wood is just like a Snickers bar. It's like yum, yum, yum. All sorts of sugar there. And so it just yields glucose. Same glucose we've been talking about. And so you get these long fibrous structures, like I said, stacking on top of one another. And it kind of looks like, well, you know what cellulose looks like. This is from an algae, and you can see like the long flat structures on top of one another. And so you see these bonding patterns. Now, one of the really interesting things about carbohydrates is that they're responsible for our different blood types. So red blood cells have chemical markers on the exterior, and one of them has these oligosaccharides on them. So remember, oligosaccharides are between 3 and 9 monosaccharides put together. And the four different sugars that are involved are galactose, a new one we call fucose, and fucose is unique in the fact that it does not have a hydroxide on carbon 6. You can see it's deoxycarbon 6. There's no hydroxyl group on it. And it's also at a different orientation. It's pointing down in the ring rather than pointing up. And then there are two new ones which we need to learn about called N-acetyl glycosamine. Why is it called that? Well, we have a galactose group here on top, and let's look and see what we have on the bottom. So we've got an N linkage, so we know we've got an amine. And what's linked to the amine? We have what looks like an acetic acid group, because we have a methyl group, we have a carbonyl, and we have lost the hydroxyl and linked it to the amine group. So we call it N because there's something linked to the nitrogen, an acetyl group, and the rest of it is galactose amine. So that is one of the sugars. That's an amino sugar because it has an N group attached to it. So those are the sugars involved in making the chain involved in red blood cell type. So type O just has a very short chain. It has the N-acetyl glucosamine followed by galactose, followed by fucose. That's type O. So it has the shortest. It also has the basis of everybody else. So you can see that all the other groups have the same three sugars attached to them, but A and B have different attachments. So A has another N-acetylglucosamine, and we call it NAG for short, because we see it in a lot of complex carbohydrates found on the outside of proteins. Proteins generally protect them, a lot of proteins protect themselves from being degraded by having a layer of carbohydrates on them that prevents enzymes from coming in and chewing them up. And one of the most common sugars in there is N-acetylglucosamine. So type A has the same group to start with, NAG followed by galactose followed by fucose, and then it has another NAG attached to the galactose. That's A. And then when we come down to B, which is fairly uncommon, type B, it has NAG, galactose, and fucose, but it has another galactose attached. And then AB, which is really rare, has both A and B present on the same blood cells. So A and B would have both of these groups present. So what does that mean for blood donation and blood acceptance if you're in the hospital and you need a transfusion? Well, the body can only recognize its own carbohydrate set. It sees that as being the proper set you should have. And so if it comes in contact with any other form, it'll try and take those blood cells out. Your immune system will recognize that if you have A, that's the one that's supposed to be there. If you're transfused with A, that's fine. But if you're transfused with B, that looks different, and it'll go after it. So because the trisaccharide on O is the same as all the other trisaccharides on O, A, B, and AB, it's considered the universal donor, because all body types recognize that as, well, that's fine. It's got the basis of all the other groups, and so we call that the universal donor. So O blood can be given to anyone, and it's fine. Your body will not reject it. On the other hand, the AB blood type would be considered the universal acceptor, because AB contains all possible AB and O combination types. It has A, it has B, and O is already present because it has those same three sugars. So if you are AB, good news for you, because you can basically get any blood transfused into you, and it's all good. So to wrap things up, so A has this particular group with NAG on the top. So it can receive other A, and everybody can accept O. And it can donate to A, and it can donate to AB. If you have B, you have galactose on top of the universal 3. So you can donate or receive B, and of course you can receive O. You can give to B and AB, but you can't donate to O, because remember, O only has the 3. O only has the universal 3. And so it can't accept A, B, or AB. That would not go well. But, so O can only get blood from other O. And AB people, they're really lucky, because they can receive blood from anybody, but they're not expected to donate to anybody either, because AB is really rare. And so, and since AB can accept any form of blood, there's not generally a really big demand for AB. There could generally be a demand for A and for B, but not for AB. Okay, so that is the story of blood types and how they fit together with carbohydrates. I'll talk to you on Tuesday. All right, bye-bye.